Antihistamine drugs, pharmaceutical chemistry 1. When we are subjected to trigger for allergic reactions, which represent a foreign entity to our body, such as antigen, a series of symptoms start to develop. This trigger could be pollen grains, insect sting, or even certain material, or even certain types of foods. Sometimes during a specific season, and viral or bacterial infections such as common cold, flu, and Haemophilus influenza. The mast cells are strategically positioned at environmental interface, such as skin, lung, and gastrointestinal tract. For skin, it causes itching, and for lungs, bronchoconstriction, and gastrointestinal tract produce more acids. Histamine is a crucial chemical messenger in the body, vital for immune responses, specifically during allergic reaction. It's a mediator for hypersensitivity and distributed in mast cells in all peripheral tissue. Histamine is released from mast cells after decarboxylation of amino acid histidine, which convert the histidine to histamine within the mast cells, then released from mast cells in a process called granulation to act on histamine receptors H1, 2, 3, and recently discovered histamine receptor 4. Within normal levels of histamine, it acts as a part of immune system, but in certain allergic reactions, when it increases upon certain levels, it causes manifestations such as itching, urticaria, and other allergic-related reaction within our body during action of histamine on its specific receptors as mentioned H1, H2, 3, and 4, and they are distributed all over the human body. Histamine 1 is located throughout the body and the most abundant receptor, specifically in smooth muscles and vascular endothelial cells, heart, and CNS, while H2 histamine receptor 2 is mainly abundant in gastrointestinal tract, while histamine 3 abundant in CNS, central nervous system, and actually all of histamine receptors present in the central nervous system. And the most recently discovered histamine 4 is dedicated for hematopoietic system and the immune system. Now we can take a look to different generations of antihistamine drugs. The term antihistamine drugs refer to drugs that block H1, histamine receptor 1 only. Antihistamines compete with histamine for its binding sites at the receptors. However, antihistamine cannot remove the histamine if it's already bound, and subsequently can be used for treatment of different allergic reactions such as allergic rhinitis, conjunctivitis, allergic dermatological conditions, and etc. Blocking H1, which is present in CNS, like mentioned previously, resulted in adverse effect, CNS adverse effect. And this adverse effect is associated mainly with the first generation antihistamines. Histamine has a role in producing excitatory neurotransmitters, so blocking H1 in CNS resulting in sedation effect and other adverse effects related to CNS inhibitory action. Antihistamine drugs can be classified into three generations: first, second, and third generation. Regarding first generation H1 receptors, which as mentioned before has highly adverse effect on CNS, the structure activity relationship involved presence of two phenyl rings which are essential for activity for blocking H1 receptor, and these two aromatic ring attached to atom X. This atom X can be oxygen, nitrogen, or carbon, and actually this classify the first generation to subclasses into ethanolamine, ethylene diamine, and alkyl amines. The structure activity relationship contains also a flexible spacer, usually 2 to 3 carbons, and at the far end there is tertiary amine that mostly dimethylamine or this tertiary amine involved in piperazine ring. This arrangement mimics histamine, the natural analog, the natural substance, but at the same time block the receptor, bind to the histamine receptor by its similarity to histamine, and block the receptor in competitive effect. The first subclass to first generation amino alkyl ether, ethanolamine, which present in blue inked in the first drug diphenhydramine. Diphene contain two phenyl rings attached to ethanolamine. The second one, clemastine, contains two stereocenters. Okay. The third drug, dimenhydrinate, and it's marketed as a Draminex. It's composed of diphenhydramine, the first one, and 8-chlorothiophylline. The role of 8-chlorothiophylline is stimulatory effect. It's similar to caffeine, has a stimulatory effect to oppose the inhibitory or sedation action of diphenhydramine on the CNS. All of the three drugs has impact on CNS. The second subclass, ethylene diamine, which contains two nitrogen atoms, ethylene diamines, such as pyrilamine. This drug contain one phenyl ring and the other ring is pyridine. From these structures, we can conclude structure-activity relationship regarding the two aromatic ring. Two aromatic ring can be two of them phenyls or one phenyl and the other one is pyridine. It's important to be non-coplanar to each other, and the para substitution enhance the activity. Another subclass of the first generation, alkyl amine, in which the nitrogen or oxygen atom is replaced by carbon. So we have 3 carbon at spacer. This modification enhances the lipophilicity and subsequently enhances the CNS effect and shifting activity to CNS stimulation by acting as selective serotonin reuptake inhibitor. We can notice from the three structures for these three drugs, they have similarity to zimaldine, the selective the selective serotonin reuptake inhibitor drug, located on the far right down of the slide. This drug sometimes abused in the pharmacy for its effect on the central nervous system, and the stereoisomer form is important for activity, in part much more active in S enantiomer for the Avil, the first drug, chlorpheniramine the parachloro enhanced the activity, and the last one, tripyrrolidine, should be in the E-isomeric form, and this form is related to binding to histamine receptor. The two phenyl rings can be fused into tricyclic system to result in promethazine, methixine, and cyproheptadine. The nitrogen atom in promethazine resulted in potent antiemetic and anticholinergic action. By substituting nitrogen atom of phenothiazine ring by sp3 carbon atom, this results in moderate H1 antagonist and marked anti-muscarinic activity, shifting the activity into anticholinergic action. And by substituting this sp3 carbon by sp2 carbon, this results in antihistamine agent and additional activity as blocking the serotonin receptor as well. Finally, regarding first generation, as mentioned before in structure-activity relationship, this tertiary amine can be involved in piperazine ring. As a piperazine derivative, cyclazine has antihistaminic action and can be used for treatment of motion sickness, and it's used during general anesthesia and opioid treatment. The second generation antihistamines rely on introducing more bulky polar groups that hinder the access to the central nervous system and subsequently more selective to the peripheral histamine 1 receptors. They have a relatively prolonged action due to their enhanced binding to H1 receptors, so they have slow dissociation. Additionally, they mostly have active metabolites. As we can see, the second generation, such as loratadine, terfenadine, and cetirizine, contain large group and large hydrophilic group that prevent access or lower the access to central nervous system. Loratadine can undergo metabolism and give active metabolites. Terfenadine withdrawal from market due to side effect, cardiac arrhythmia side effect, but with oxidation at this position result in active metabolite. Cetirizine is dextroisomer and its levoisomer is also active drug. Now we can go for the third generation. They are derived from the second generation by metabolism or their active enantiomers, such as levocetirizine. This drug is the active enantiomer of cetirizine, only different in the chiral carbon, the levo form of the dextro form cetirizine. They took the active metabolites and used these metabolites as drugs. The active metabolite of terfenadine, called fexofenadine, is more effective and safer and marketed under the brand name of Telfast. Also, loratadine has active metabolite desloratadine by hydrolysis of the amide bond. And this compound, desloratadine, is marketed under the brand name of Arius. So we have second generation as example of three presented drugs, and the third generation that produced from the metabolism or the active enantiomer form. Thank you for listening, and thank you again for being here. Thank you.